Welcome to today's Life Sciences lesson. My name is Griselda van Wyk, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Centre. Yes, and today we will be looking at paper 1. Just a short overview of paper 1. Obviously, we cannot do the whole thing because that will take a year. Right, so first, let's start at the content weightings. When must you start studying? I hope that you already started with responding to the environment. Why am I saying that? Look at that. 54 marks of tomorrow's paper will be about the nervous system. Right. So I hope that you already know the nervous system, the eye, the ear, the brain, the reflex arc, and all those neurons, and that you also already know human reproduction. Because look at human reproduction, 41 marks. That's the second biggest topic of tomorrow. So between these two, you're already at 95 out of 150. Okay. But what is left then is the endocrine system. That's 34 marks, right? That's all your hormones and things. And then plants, the hormones of plants is 13. And lastly, you will study the reproduction of vertebrates. So that's the order in which I recommend that you approach your studying. But like I said, by this time you should have already done the nervous system and at least human reproduction. And you are taking a break now with me just to see are you on the right track. Okay, so let's go. Firstly, remember tomorrow, I hope you know this by now, the paper will be out of 150 marks, 2.5 hours. There will be two sections, a section A and a B. Section A is basically your multiple choice questions. You will have terminology questions here and that column 1 and column 2. And all of it will be 50 marks of the paper, right? A third of the paper. This is the easy part of your paper. Then section B will be 2 questions. You'll have a question 2 and a question 3. And this is where they usually give you a diagram with questions on the diagrams, or they give you graphs, practical investigative questions, and so on and so on. So you'll have question 2 will be 50 marks and question 3 will be 50 marks, right? And that will give you 100, and together it will be 150. Right, so what do you have, or what do you have to your disposal in Gauteng province? Right, firstly, we have done a lot of broadcasting lessons throughout the year and last year. It's all on Google Drive. And your teachers also send you a page with all the links that you can just click and you can see nervous system, or you can see reproduction, or whatever that you need help on those links. Right. So they are there for your disposal. If you're alone tonight and you don't know who to ask, those lessons will help you. Right. Then web lessons and voice notes. We sent you, all the teachers, we as Gauteng department sent all the teachers the voice notes per topic, and they were instructed to send it to you via WhatsApp and email. But if you do not have it that way, you can also use the Google Drive. Now your teacher was supposed to send you these Google Drive links, and every single subject in grade 12 in Gauteng has a Google Drive link, and on this link you will find this broadcasting lessons, the web lessons, the voice notes, topic tests that you were writing this year. There's even question papers and so on per topic. Right, so this Google Drive link is very important. If you do not have it, please make sure to contact your teacher. They must email it to you or get it to you so that if you're not sure, you have that link. Right, another very important thing today to do is to work through past papers. That is the most important thing that you can do now. After studying, you do your past papers, especially your end of the year papers, the June DBE papers, and also other provinces prelim or previous year's prelim papers. Right. Then you've got your exam guidelines. People, that is so important. Remember, you have to tick off as you go through your exam guideline. I finished this topic, I finished this subtopic, and so on. That's how you know that I've done everything. Right. Textbooks is also important. And then mind the gap. Oh, mind the gap is also very nice. And your SIP material, we sent all the SIP material up until the end of term 3 to you guys, as well as the DBE study guides. You don't have to look at all of them, but if you need resources, it's all at your disposal. Okay, so I hope you have it. Let's look at marking principles. What do we as teachers look at in grade 12 when we're marking your script? And especially at the end of the year. But your teacher is also supposed to do it when they are marking your test after tomorrow. If they ask a question that gives 3 reasons, but you give 5, and say, for example, now your first 3 answers is wrong, but your 4th and your 5th answer is correct, the teacher is not allowed to give those marks. The teacher must only mark the first 3. So do you get what I'm saying? You must think very carefully before you answer and make sure that the ones that you are very sure, you start with them, right? Because we only mark 3, the first 3. Right? If they ask comparisons, you have to compare 2 structures or 2 processes or whatever, and you just give a description of the process or the structure, we will not give you marks, except if you say this one is like this, which is different than that one. Then there's some kind of comparison there, and then we can see, okay, no, there is some comparison there, and then we'll give them mark. So just look at what we are asking in the question paper. If tabulation is required, you must table the data or whatever, you need to draw a table. And please people, I know it's only 1 mark, but 1 mark is 1 mark. It can be the difference between failing or getting a distinction or whatever, right? So you tabulate. If they ask to table, you do your tabulation because that's 1 mark. Flow diagrams. People, we don't ask flow diagrams in life sciences. Never, ever, ever draw a flow diagram. So if we ask you to explain a process and now you start drawing a flow diagram, we are not allowed to mark it, right? Even if the facts are correct, we are not allowed to mark it. So please do not use flow diagrams. Yes, you do it when you study. You can use your flow diagrams and study from them, but you're not allowed to draw them, right? Then, if you have a question where there's letters and so on, and we ask for the letter that does this or that, and you give the name, we cannot accept the answer. Even if you are correct, but you didn't answer the question, you must answer the question. The same if we ask the name and you give us the letter. Even if it's a correct letter, it shows to the correct structure, sorry, we cannot give you that mark because the question was asking for the name. So make sure that you read the question. Right. So let's start with human reproduction, which is 41 marks, the second biggest topic of the paper tomorrow. And as you can see, we have female reproductive system there. You'll have to look at the reproductive system and the ovum there, male reproductive system, the whole reproductive system with the sperm cell there. What is puberty all about, right? What is causing it in males and females? Menstrual cycle, that whole event, you know, it's a big thing. We'll go through that this afternoon, as well as the hormones that is part of this menstrual cycle. And then lastly, the fetus, the development of the fetus, you know, the amniotic sac and all those things. Right, so let's start off with the male reproductive system first. People, we can ask it from the front and the side. I know that usually we do it this way. Nay, we give you that frontal view and you know it, right? But now we can ask it from the side as well, and you mustn't get confused. I think the thing that confuses learners the most is the bladder. The bladder is very big. Can you see? The bladder is very big. Look there at the side view. It's very big. And then you can see the prostate gland is very small in comparison to the bladder. So don't confuse the two. Okay. So obviously you need to know your labels there. Where's the prostate gland? Here's the Cowper's gland. This is the vas deferens, the epididymis, the testis, the urethra, not ter. Ter is for the bladder. Right. Urethra. And the seminal vesicles is just above the prostate gland. If you look on the side, you can see here's the structure. There's your testes with your epididymis, the vas deferens going up. And now we have the bladder here. Don't get confused with the bladder. It's there. It must be there. You have to have a bladder. It's just below it. Well, you'll find the prostate gland, and then your urethra will go there. Okay, so don't get confused there. But you need to know every single label and also the function of them, especially of the accessory glands. Right, that's those 3 glands there. Let's see, I've got a slide with only the accessory glands on it. The seminal vesicles, that looks like little leaves, they produce seminal fluid to transport, they help transporting the sperm cells. Right, then the prostate gland secretes clear, slightly alkaline fluid that now neutralizes your this acidic environment in the vagina, and that's about up to 1/3 of the volume of the semen. And then also Cowper's glands secrete fluid containing nutrients for the sperm. So the prostate and the Cowper's gland will nourish the sperm, enabling them to swim. And another tip I just want to give you, just go to mind the gap and look at mind the gaps function of the accessory glands. Okay. Then the female one, the female reproductive from the front, very easy. You can see it looks like a little lampshade. There they are. There's the ovaries, the fallopian tubes, and this will be the vagina. Your knees will be the cervix. Your this will be your uterus, sorry. Your knees will be your cervix, and your vagina will be the bottom part of your legs, right? And your vulva will be your feet. You can, so it's like a little, you know, you sit in the exam hall tomorrow and you just sit like this and you think what is what. It's an easy way to remember it. From the front, yeah. So the uterus is the top part, yeah. That's usually where the baby will be, when the baby grows, right? And then the ovaries is this part, yeah. And this is your fallopian tubes, like I said, your little lampshade there. And the vagina is at the bottom, and there's your cervix, right? And you need to know what the function of them is, and so on. But now look from the side. That's hectic, So firstly, remember, if you look from the side, the front part, yeah, will obviously be the bladder. The bladder will come first. So don't confuse the bladder now. There's the bladder, right? That will be your first hole. The second hole there will be your vagina there, and then your uterus will be there, and there's your ovary. Yes, I know. From the side, it becomes very difficult. And this is obviously the intestines, right? So yes, you need to know your stuff if you ask it from the side, because you are used to this way of asking. We usually ask it from the front. Right, and puberty. Now, you all know what puberty is. You just went through it now. Some of you are still going through it, So you'll see that when you're a male, you'll get a beard, you'll grow big, muscles, you will have a deepened voice, and so on. A lot of hair, spray a lot of deodorant, you can smell you from a mile away because it's just deodorant, and so on. The females also hair, breasts, and so on. So this is all started by your hypothalamus, right, which secretes the GnRH hormone, which you do not have to know, but you need to know this. In males, puberty is caused by testosterone, right? That's a male hormone, and it's causing all these things in boys. Why they become so crazy. And then estrogen, yes, that's the one in females that causes all the, the breasts forming, the menstruation, all that stuff. Right, so now we're going to look at gametogenesis, and this you need to know. People, only look at your exam guideline here. Do not go to your textbooks. Your textbooks are old. They still have the old way of many things. We were talking about lots of stuff there, lots of steps and so on. No. Go to your exam guideline. It's 4 lines for spermatogenesis and 4 lines for oogenesis. That's all you need to know. It's 4 lines. If they ask it, it's easy. You just write it down. You don't even have to think. It's plain and easy. Right. In the olden days, we had a very, very difficult, complicated system that we taught the learners. Now it's very easy. Stay with the exam guideline here. Right. And this processes, this gametogenesis, the spermatogenesis gives us sperms. Obviously that will be in the male. And in the female, oogenesis will give us a ovum. Right. So that's the end product of that processes. Right, let's look at the ovum first. You need to be able to recognize this structure and label it. Right, so firstly, we'll have the outer layer there. You can see it's irregular. Right, it's very irregular, and that's the corona radiata. Then inside this part here, that's easy. It's the cytoplasm. The nucleus, remember the nucleus is always haploid. This is after meiosis. It's haploid. Right, we have a haploid nucleus because it has to fuse with another haploid nucleus to give us zygote. That's diploid. So it's a haploid nucleus. And then this layer here is the zona pellucida. Right, so the nucleus bears the genetic material from the mother. Obviously not the father. The father's not the one giving the ova. The mother is. Right, and we're going to go through the process of how this happens. And then the vitelline membrane, which is this part here, it's basically transparent. That membrane there is transparent. Right. Now the sperm. Oh, they love to ask the sperm as well. They will maybe ask you to draw it, or they'll give you a sperm, and then you have to talk about the structural and the structure and the function. Right, explain that. So an easy way also to remember is take your hand like this. Right, so this part here will be the acrosome, and this will be the head where the nucleus is and the cytoplasm. And this is where the middle piece where the mitochondria is. And then here is the tail. So there's your little sperm cell going around. Why do you need an acrosome? That's for the enzymes, right? So the enzymes that digest the outer layer. And you need the nucleus because that's a genetic material. The mitochondria is there to give energy for this sperm cell to get that swim on, because it has to swim all the way through the vagina, up to the uterus, through to the fallopian tubes. It's quite far for a little guy like that, so it needs a lot of energy. And then those tails are the ones now, the swimmers, right? The one with the best tail can get there the fastest. And yes, I think I said all those things there. The long tail, so if it's got a long tail, it will be able to swim up to the sperm and fertilize it, right? Then let's go to the very complicated process of menstruation. This is a very complicated process. Don't you feel sorry for us, the females? We have to go through this every month. It's terrible. Anyhow, let's see what's happening. The ovarian, you need to know the ovarian and the uterine cycles, right? That's very important. Ovarian is the ovums. Uterine is the uterine lining. What's happening there, right? So, we're going to start here. Now, the first part of the month, you will see there's menstruation happening. The lining is being shed. It's been shed and it moves out, right? So, the uterus cycle, the lining breaks down and it's released. But other things are happening in the body now. Firstly, the ovaries. New follicles are starting to develop. Can you see those little spots there in the ovaries? They're becoming bigger. That's a follicle. It's starting to develop, right? Because things must happen now, right? And all this is done or stimulated by the pituitary gland, which is in the brain. You remember the brain? It's there. And it secretes follicle stimulating hormone. If ever name told you what it does, follicle stimulating hormone. It's stimulating the follicles, right, to grow. Now, in day 8 to 13, you'll see there, can you see the uterine lining is now becoming thicker? Why is it becoming thicker? Because the ovaries here, that developing follicle, which we call the developing follicles, as well as the biggest one, the Graafian follicle, secretes estrogen. Right, estrogen. And estrogen now will stimulate the uterine lining to become thick and granular. It grows, right? So it has an influence on the uterus. Next part here is from day 14, where the pituitary gland, the brain, is doing something again. It's secreting LH, luteinizing hormone. Luteinizing, causing a corpus luteum to form, right? So this stimulates ovulation. Can you see there? Look at that little thingy there. That's ovulation, people. And now the race is on. That little ovum is in the fallopian tube. So now it's waiting for a little sperm cell to come and fertilize it. Right. So everything is waiting now for this moment in time. Because if it's fertilized, a baby can grow. Right. But usually that doesn't happen. But anyhow. So the Graafian follicle, remember that's the biggest follicle, the one just before ovulation, bursts open, releases the ovum into the fallopian tube. So now she's moving and she's looking for a sperm. Right, now what happens? Day 15 and 22, that is now in the ovaries. This Graafian follicle that burst open is becoming a corpus luteum. Right, and this corpus luteum is secreting progesterone. Right, so that's the first thing you must know. And what does progesterone firstly do? It thickens. It stimulates the endometrium, this lining here, to become more thick and glandular, blood vessels and glands. So if fertilization takes place and there's a little embryo implanted, that this little embryo can be safe and get nutrients. Right, so that's the purpose of that. Then, oh yeah, I forgot to say this. The progesterone here is very important. It inhibits the pituitary gland here from secreting more FSH. You don't want for more FSH now, because what will happen? While this little ovum is now running around there and swimming around there in the fallopian tubes, what is happening? Now another follicle is being formed. So if this one is fertilized and the next one is fertilized, you're going to have a problem, right? You're going to have babies at different ages and it's not going to work, right? So immediately the progesterone will inhibit FSH from stimulating another follicle. So there's only one ovum is there, ready to be fertilized. Right, now, the next phase, there's two things that happened. Firstly, did fertilization take place? Okay, no. Did it not take place? So there was no sperms. No sperms there, nothing. Then this corpus luteum will start to shrink, right? And stop secreting the progesterone. So the progesterone levels will start to fall. If fertilization took place, so there was sperm in that fallopian tube, right? The corpus luteum will keep on now secreting that progesterone to keep this uterine wall thick and glandular, right? But no more follicles will be developed and no menstruation will take place, right? So that is the ovarian and uterine cycles in a nutshell. But I like this way. We've talked about it now, pictures and everything, but this graph shows you everything you need to know. So let's start. Yeah. So you can see at the bottom here is the uterine lining, the uterine cycle, ovarian hormones, obviously those hormones that's here in the ovaries, the pituitary gland hormones, then your body temperature, and the follicles. Right, the ovarian cycle. So let's start in the beginning. So what happens first is the pituitary gland will start to secrete follicle stimulating hormone, which will then impact on the follicles in the ovary. Look at them. They are now developing. And what do these developing follicles do? They secrete estrogen. Can you see there? The estrogen levels are going up. And what does this estrogen level do? It will increase the size or increase the thickness of the uterine lining or the endometrium. Before I go on, obviously the first 6 days or 7 days or 5 days, there will be menstruation. Right, you can see here. Right, that's when the previous cycle is now finishing up. And so we get rid of that and now it's starting again. Right, so now this uterine lining is now nicely growing because of this estrogen there. And then just say between day 13 and 14 and 15, depends on everybody, on the individuals. You can't say every individual day 14. Some will be day 12, you don't know. There will be a sudden spike in luteinizing hormone. And this is now secreted by your pituitary gland. So if you get a graph and you just see this little plump, it's like a spike, then you know this is LH. And LH causes ovulation. There you can see it. So the Graafian follicle now bursts open and an ovum is secreted. Right, and you can see the estrogen falls down. But now what happens is a corpus luteum is formed. And what does a corpus luteum do? It will secrete progesterone. Look there. So the progesterone levels go up. And remember what progesterone does? It will inhibit FSH. You don't want any other follicles now moving around in this game, because it's all a game now. You don't want no other players here. Only one player. Right. So this progesterone is now inhibiting the FSH. So no more follicles are being stimulated to develop there. Right. And another thing this progesterone is doing, it is increasing the thickness of your uterine lining or your endometrium. It's becoming more glandular and more vascular. Can you see there? So it's become thick and a lot of blood supply in case implantation takes place. But in this case, you can see nothing happens. That ovum was there and then gone because there was no sperm. So this corpus luteum will start to shrink and shrink and shrink away. Right. And as it's shrinking and disintegrating, what will happen? It will stop secreting progesterone. Can you see that the progesterone levels are decreasing? And when progesterone levels decrease, what happens to the uterine lining? It will also decrease. Menstruation will take place. Right, so this is when fertilization did not take place. But now, if it did take place, if there were sperms in that fallopian tube, everything up to here is exactly the same. Nothing has changed. Right. But now something happened in that fallopian tube. That little ovum got a sperm. Things happened. Right. First thing you look at is the corpus luteum. Can you see it's the same size? It does not shrink. It does not disintegrate. It stays the same size. That's the first thing you look at. The second thing you look at is the progesterone. Look at that. Because it keeps on secreting it. Can you see? So your progesterone levels stay high, right? And then the third, so this is the first thing you look at. Then the second thing, the progesterone levels. The third thing you look at is the endometrial lining. It stays thick. There's no menstruation taking place because progesterone will maintain it, right? So that's the difference between fertilization when it happens and not happens. Right, and this is also a nice flow chart. Just remember, you're not allowed to draw a flow chart in your paper, né? And this is just a summary of it. So it all starts with your pituitary gland, which secretes FSH, follicle stimulating hormone. This stimulates the follicles to develop. What do? They secrete estrogen. There's the second one. Eh? Estrogen causes the thickening of the endometrium. Now also the pituitary gland will secrete LH. Right? What does LH do? It causes ovulation, right? Which then forms a corpus luteum, which secretes progesterone. So if you can remember FOLP, you can remember the sequence in which these hormones are secreted and what they do, right? Because that is important. Right, now fertilization has taken place, and you know there's no things happening there in that fallopian tube. Firstly, there you can see is our little ovum, and there it is being attacked by those sperm cells. Fertilization is taking place, and remember, only one can go in. The moment that first one has gone in, that membrane closes up, it freezes up, right? So no more sperm cells can then penetrate. Okay, so this process here is fertilization there. Now we have two nuclei that fuses, right? And then they start to divide. And this process here is no longer now meiosis, it's mitosis, because now the cells are just doubling, it's growing, right? Mitosis. So this is mitosis, right? And then you get your morula, that's a ball of cells, and then you get your hollow ball of cells, which is the blastocyst. So there they go, it grows, and there's our hollow ball of cells, the blastocyst, and that is the one that will implant in the endometrium. Right, and then this is the result. There's the little fetus sitting there, and you can see that this is now the uterine wall, yeah, over here from the mother, right? And we call this whole structure here the placenta, because the chorionic villi from the chorionic membrane, the extra embryonic membrane from the embryo, will then infiltrate the uterine wall, and you can see here there's intravenous spaces and everything, and this will then be bathed in blood. Remember that the blood of the mother and the baby will never mix. It doesn't mix, but there will be blood there, and through the diffusion, the baby will get oxygen, or the CO2 and the metabolic waste from the embryo, I must say embryo or fetus, will then go to the mother so that she can excrete it through her kidneys and so on. Right. But you must know the following. You must know the amnion, which is this structure here, right? And then the amniotic sac and the amniotic fluid. Remember, the fluid is where the baby is in. It's like a little shock absorber and so on. The umbilical cord. There you can see the umbilical cord. And it's the arteries and the vein. And what else did I miss here? Let's go to the next slide, because that's a very nice summary of this. This is a lovely summary. So firstly, when you look at the summary, you'll see there's a placenta over here, right? Structure here. It's oval, it's vascular, meaning there's a lot of blood there, blood supply there, and it nourishes the uterus. The function of it is gaseous exchange. Like I said, the O2 and the CO2 between the mother and the baby is exchanged through diffusion, right? And nutrients from the mother will go diffuse into the baby via the umbilical cord, and this also secretes progesterone, right, to help to keep this layer intact. So it takes basically the function of the corpus luteum over after a while, and it secretes progesterone to maintain the pregnancy. Right, the chorion is a extra embryonic layer, right? And you will learn later or hear later about in the amniotic egg where this also comes in, right? And it basically, this it covers, it's the outside, it covers the amnion, right? And it forms the chorionic villi, which I said this part here, that forms the placenta, right, from the baby side. Then the amnion, this is a structure. It's the innermost extra embryonic membrane. It secretes amniotic fluid. There it is. And that will help protect the baby or the developing fetus. You must not say baby, you must say fetus. Amniotic fluid, this is the fluid in here. Imagine there was no fluid. Now you're walking around, this poor baby would bounce in here, hit his head against your hip bones and whatever. Shame, man, it would be terrible, right? So we need that for shock absorption, right? So that's a function of this amniotic fluid. It also protects this fetus against temperature changes. It keeps the temperature nice and, nicely there. It's like the aircon in there. And then against any desiccation is drying out. The baby doesn't dry out. It's nice and moist skin and so on. Chorionic villi, like I said, this is the finger-like projections of the chorion, which projects into blood spaces of the uterus of the mother. It provides maximum contact with the maternal blood and forms part of the placenta. Right, so it's not... Remember, the blood of the mother and the fetus never mixes, right? Umbilical cord, this part here, very important part here. It's a cord-like structure containing the umbilical arteries and the umbilical vein. Now, how you can remember it, just draw a little face. 2 arteries and a vein. Right, there's always 2 arteries and a vein in an umbilical cord, right? And the arteries will carry the wastes away from the baby. So it will carry away the carbon dioxide and the metabolic waste, all the nitrogen waste and all those things, to the placenta, where the mother's blood will then, it will diffuse to the mother's blood, and then the mother's body will get rid of it. So your mother's worked very hard for you, way back. The umbilical vein carries the oxygen from the mother, right, to the fetus, right. Okay, so this is a very nice summary. I hope you can find it also in the DBE exam guidelines, or not exam guidelines, the DBE study guides, right? But also, we also made it available on a lot of our PowerPoint presentations that your teachers used. So that's basically human reproduction in a nutshell. Like I said, I didn't go into too much detail because we do not have time. The next one is human response to environment, and that is the nervous system, right? Now, this is the biggest one. Can you see? 1/3 of the paper, right? So you really need to know everything here, because it's 1/3 of the paper, people, right? So what is in this section? You've got neurons, right? So you need to know the different kinds of neurons and their labels, the brain, the spinal cord, right? And here the reflex arc will come in. And then the human eye, all the labels there, and also the 2 processes there, accommodation and pupillary mechanism. And then the ear, of course, hearing and balance. Right, so you need to know all these things. This is a big portion of tomorrow's paper. Right, so when you respond to the environment, firstly, you need to know your peripheral nervous system. Peripheral is to the sides, right, from away from your body. So you've got the central nervous system, which is your brain and your spinal cord. Then you get your peripheral, which is your cranial nerves from your head and from your spine going outwards to your arms and your legs and so on and so on. Right, and then your autonomic nervous system is what it says. It's automatic, right? You don't have to think about it. It just happens, which is lucky, because some of you, if you had to think about breathe in and breathe out, you'd be dead already a long time ago. No. So this is happening automatically. You don't even have to think about it, right? And here you got the sympathetic one and the parasympathetic one. You always just learn the one, and the other one is the opposite. So if we think about the sympathetic one, this is the one when you're in stress. Like tomorrow when you're in that exam, or you are stressed out, né? Or you're being chased by a dog or a lion or whatever, what happens? Your heart rates will increase, right? And your blood pressure will go up. You will start breathing faster if you run away from a dog, right? Because you need a lot of oxygen in your muscles to get away. You won't be needing blood in your digestive system. When you're running away from a dog, you don't need to digest food. No, not at all. Your pupils will dilate. You become bigger, more light will go in, more information for your brain to make decisions on where you're going to jump or what. And also, the wall of the bladder will relax. That doesn't mean now that you will, urinate. It just means that you will not urinate now. But if it's a lion, maybe you will. Right. Now, the parasympathetic one is the opposite. So if the blood pressure will increase here, it will decrease on this side. If breathing rate increases, it will decrease. So that's the easy way to remembering. That will be the opposite. Right, if you look at the brain, very important. Please know your brain. You've got this big part, that's the cerebrum, right? This is where voluntary action is, memory, higher thought, where you think, right? You're thinking, yeah. I know some of you are not thinking, you must start thinking now, né? And you must also use this because you must remember for tomorrow. So this is the part you're going to use to remember all these facts. The corpus callosum, this is this part here, and that connects the left and the right brain, right? The pituitary gland, that little pea-like structure there, a very important gland. Your whole endocrine system is being run by this, and it secretes a lot of hormones that we will talk about later. Then the medulla oblongata, just here below this bump here. This one, if it's severed, if it's damaged, you are dead. Because look at it, it controls your breathing, your heartbeat, and it transmits all the nerve impulses from and to and from the body to the brain. Okay, and otherwise. So, your brain dead. If your medulla oblongata is at all severed or damaged, you're dead. Right, then the cerebellum, here at the back, they always call this the tree of life. It looks like a little tree there. And this is for balance and muscle tension, right? So that your muscles are nice, it can move and so on, and you have balance. So this one works with the ear as well. And then the hypothalamus, right? Yeah. Now this one is one that you guys are really familiar with because it controls your hunger. I've never seen people eat like a high school boy, right? You can eat. So your hypothalamus is very, very active. Thirst, very thirsty as well. Emotions, also a high school thing, secondary school thing. You're very emotional at that stage. Your hypothalamus is all over the place, right? And remember, we always make the joke that if you break your heart, it's not actually your heart that was broken, it's your hypothalamus that's sad, right? Because it controls your emotions and also your temperature, right? So if your heart cold and your body have to respond to that, that's all controlled by the hypothalamus, right? So you need to know these. If you don't know your book, you didn't do your work through the year, just go and look at the Mind the Gap. It's got everything there that you need to know. Right, the reflex arc. This is now, get two things. The reflex arc is the actual structure that you see here, right? The action is the actual action that happens, right? So this is to protect your body against injury. So now, if you're stupid enough to touch a candle there, no, you're not very clever. Fingers on the candle, what happens? Before you even think, take away my finger, your finger's already gone. It's gone there. Right. That's a reflex action, people. Your body is now protecting you. So what happens is, this is the receptor. It has a stimulus of ouch, it's hot, right? So it converts that stimulus into a nerve impulse. Remember, we're not allowed to talk about messages. When you talk about the nervous system, it's always impulses, right? So the nerve impulses will then go alongside the sensory neuron. Just remember senses. I'm hearing, seeing, feeling is a sense. Sensory neuron, right? And that's the one with a little bubble there. It looks like always looks like a little alien eye checking you, right? So you can see it's got a little bubble there. That's your sensory neuron, right? And it goes into the spinal cord here. And that one there is your interneuron. And between them, there's little gaps there. You can see there's a little gap in that. That's a synaptic gap. You must also know about that, right? That's where the chemicals will move the instruction or the impulse forward to the other neuron, right? So the neuron moves over the synaptic gap to the motor neuron. Motor neuron is always from the spinal cord to the muscle or whatever now needs to react. And in this case, it will be a muscle. It's the effector. It's effecting change, right? It is pulling away your arm. And remember, while this is happening, there's another neuron going to your brain. So by the time your hand is here, your brain has now registered ouch, right? But then your finger's already away, right? So reflex actions are very fast. And it's to protect your body. It's like blinking when a fly comes to your eye. You don't even think about it, you blink already. You just get that fly out of your eye. Things like that. Coughing and so on. Right, so this is just another picture to show you that in this case, this guy also not very clever, touching the warm plate there. This is a stimulus, right? So it's been converted to a nerve impulse via the afferent pathway or the sensory neuron. That's a dorsal root going into your spinal cord. And this piece here, this bubble, is a ganglion, right? So it moves there into the interneuron. But can you see there's another pathway? This one is going to the brain, but it takes so much longer, you know, to go up to the brain, and then the brain must tell you what. It takes too long. So it bypasses that there, and it goes back to your effector here. So this in this case, the bicep is told to contract, and your tricep is told to relax, right? So that your hand can move up. That is a reflex arc. Like I said, it's the structure, but the action is the actual action that happens to save your finger. Or else that finger will just keep on burning, and you will start smelling burning meat, you know, and you will think it's maybe a sausage, but it's your finger. Right, eye. Yes, of course. They will always ask an eye, people. In some way, they're going to ask an eye. You need to know the eye, right? So if you always start with the labels first, because if you don't know what those structures are, how are you going to answer any question on the eye? So the first part, this tough outer layer, white layer, is the sclera, right? And it covers the eye and protects the eye, right? And it also maintains the shape. The next part there is the cornea, right? This bubbly part in front of your eye, right? And it's transparent, it's curved, and it allows light to enter, but it also breaks the light. It will refract the light. And the most refraction takes place here. And then that little, let me just see here. You can see it's showing this dark layer there, right? The dark layer. The dark layer is called the chorioid. Now, please don't confuse this with the chorion. Remember with the fetus, there was a chorion. It's basically spelled the same, but the last part is different, right? Just think of a baby or when you're pregnant, it's prenatal with the N. So with the baby or the fetus, it will be chorion with the N. Yeah, it will be chorioid, right? And this, what is it? It's the middle layer. It's in between those two layers there. And it's continuous with the ciliary body there, the ciliary muscle, and the iris, which is the round part in your eye, the circle you can see. And it has dark pigments, it's very dark, to absorb the light that comes in. If it wasn't for that, your eye would be like a disco ball inside. And the light will come in and then it will be refracted and reflected, and it will shine out of your eyes. It would just be weird. And you won't be able to see anything. So you need your choroid, né? And it's got blood vessels to also provide nutrients for your eye and oxygen. Then the inner layer, yeah, that's almost the most important one. That's a retina. This is where all your photoreceptors is. This is the reason why you can see. Right. So if your retina is damaged, you got a problem. You're not going to be able to see. Right. So it's got the rods and the cones. Just remember, cones, C for color. Right. C for color, and then rods is for black and white. And then this is where the images form in your eye, and it converts light stimulus to a nerve impulse. And where does the nerve impulse go? It always goes to the now I can't remember the neuron. I will get to the word just later, not now. Aqueous humor is the front part here in front of your iris, that little bubble below your cornea, right? And basically, it's clear fluid that gives the shape to the cornea. And it's also filled with nutrients and oxygen. It's also keeping your eye alive and nice. And then the inside there is a vitreous humor. And this one is more jelly-like, right? And it helps to keep your eye, the shape of the eyeball. It helps to maintain that. And it also helps a bit with but not a lot, with the refracting of the rays, right? Then we get the ciliary body. That's the one with the muscles, right? And this one is now helping the eye, the lens, to change shape, right? And the suspensory ligaments, which attach to the lens and then can pull the make it less convex and more convex. And I'll go a bit faster here. The pupil is basically a little hole there that allows the light to go in. And the iris, there's the round part you can see in your eye, the colored part there. And the lens can shape can change shape to see far and near, and it's elastic, transparent, biconvex. This spot here is also like always to ask that one, the yellow spot, right? And that contains the most amount of cones, right, or only cones. So that's why you can see very clear there. And then this spot here is the blind spot. That's where the word that I couldn't get to, optic nerve. Thank you very much. Oh, old age here. My cerebrum is not working nicely. It carries those impulses, those nerve impulses to your brain, right? Then accommodation, people, you need to know that when you look near or far, your shape of your lens will change, right? For near vision, when you look closer than 6 meters, the first thing will happen is the ciliary muscle will contract. And when the ciliary muscle contract, the suspensory ligaments will slacken. So they will slacken. So here's the muscle. It's contracting. It's becoming big, right? Very big. Because when a muscle contracts, it becomes big. Takes up a lot of space. So now these suspensory ligaments are slackened, right? So they don't pull on this lens so much. So this lens can just plump down and be more convex, right? Or more, but we're not allowed to say more round. Nay, we're not allowed to say that anymore, but more convex, right? So the tension will decrease on the lens and will become more convex, right? So for far vision, it's just the opposite. If you need to look far, a flatter lens will be better. But now you can't say flatter, you must say less convex. So what happens is the ciliary muscle relaxes, and when it relaxes, it becomes smaller. So it's smaller, now it takes up less space, so it's pulling on the suspensory ligaments to become taut. So the suspensory ligaments are now taut, it's pulling on this lens, so this lens can't be like this anymore. It pulls it and it becomes less convex, right? So the tension will increase on the lens, it becomes less convex, right? And then the refractive power will decrease, right? And why do we need that? If you look at the next slide here, you will see that's what I was talking about, the suspensory ligaments being slackened or taut. If it's taut, it pulls on that lens. If it's not, it doesn't pull so much, right? The tension will decrease. And when you look at a close by object, you need a more convex lens. But if it's flat or far, you need a flatter lens or less convex, because it breaks the light less, right? But you don't need to know that part, just the other part I showed you. Pupillary mechanism, people, this is very easy. Just look at your eye in the mirror when you study this. You've got an iris there. It's a circle. What does a circle have? A radius, right? So these long things here you see here is the radius of a circle. And then the other muscles are circular muscles because it's a circle, right? Do not confuse it with the ciliary muscles. This is circle, it's a circle, circular muscles and radial muscles, right? And then also easy way to remember, I will always start here. If you're in a very room where there's not a lot of light, right? So your pupil needs to be big, right, to get all this light in. So what happens is the radial muscles will contract. When they contract, they become big and short, right? You can see it big and short. So they're basically pulling up the pupil, that inner part of the iris, né? So the pupil becomes bigger, right? It becomes bigger. But if this one is now contracting, the other one will relax, right? So this is, we've got this little mnemonic here. So the circular muscles will relax. The radial muscles, those little radius muscles there, will contract. The pupil will dilate. Dilate becomes bigger, and then more light will enter your eye. And the other way around, if you have this. Here you can see your radial muscles, they are small. Né? They are small and long because they are relaxed. And when they are relaxed, they become longer. So they basically push the inner part of the iris down. Né? So this pupil becomes constricted, the radial muscles is relaxed, but if the radial muscles are relaxed, the circular muscles are now contracted. Okay, and then less light will go through. Phew. And then the difference here, you need to know the difference here, people, between accommodation and pupillary mechanism. The stimulus in accommodation is near far vision. Pupillary is intensity of the light. Parts involved here will be the lens, suspensory ligaments, and the ciliary muscles. In light, it will always be your iris. with the radial muscles and the circular muscles, right? Then what happens here? The shape of the lens will change. Here, the diameter of the pupils will change. And what bring about these changes? The suspensory ligaments, ciliary muscles, radial and circular. So, you need to study that, people. The structure of the ear, you need to know the structure of the ear. I'm not going to go through it because time is against us. The outer ear, you must know that the pinna will trap the sound waves. It will go to the tympanic membrane, which will then transfer it to the ossicles in the middle there, in the middle ear. So you know your three ossicles names: the malleus, the incus, and the stapes. The hammer, the anvil, and the stirrup, right? And then, but we actually prefer the scientific name. So this malleus, incus, and stapes is perfect, right? What's the function of the ossicles? To amplify these vibrations because it's air, right? And then, just to show you how, because the tympanic membrane is much bigger in surface area than your oval window, that's why the sound will be amplified. Also, your malleus is much bigger than your stapes, so the sound is amplified from big to small. All right. Then the eustachian tube, very important function here. It will equalize the air pressure on both sides of your tympanic membrane because it opens up here in your throat, right? If you have excess fluid, which you shouldn't have, but if you have fluid in your middle ear, it can drain through your throat, right? So that's also a nice function. Here you can see, you must know the difference between when you go up in an aeroplane, the air pressure becomes less, so there's less air pressure in here in your auditory canal, but in your middle ear there's more. So now your ears start to pain. That tympanic membrane is now being pushed out right from the middle ear. So what you do is you just open your mouth and you do this so that you can force some of the air out of the middle ear, right? And then the air pressure on both sides of your tympanic membrane will be the same. When you go scuba diving, like can you believe it's me that, ne? Yes, it is. And if the people who do scuba diving will know my buoyancy is perfect there. But anyhow, when you go down, the pressure becomes higher. So that water pressure there is hectic, people. Now it's pressing on your tympanic membrane. It's pressing it in. So you also now need to blow on your nose, force air into that eustachian tube, into your middle ear, so that the pressure can be equalized on both sides. Right, the inner ear, please know the structures here. I'm not going to talk about that too much now because of time. You need to know your structure of your ear and all the labels and functions there. And then the path of the sound, people, big thing here is that you need to know that on this side here, we are talking about sound waves. From here, the tympanic and your inner ear, it is vibrations. And then inside your cochlea and here, it will be pressure waves. And from there on, it will be a nerve impulse, right? So just know the order in which it takes place. And also in your ear, semicircular canals contains the crista, that's for balancing. Sacculus and utriculus contains the macula, that's for position of your ear. And your cochlea has the organ of Corti for hearing. And this is what I said, this is a very important slide. You always have sound waves at the start, then it goes over to vibrations in the middle ear, to your oval window. This sets up pressure waves, and in your organ of Corti, it is then converted into a nerve impulse to your brain. And yet you can see the amplification of the sound. Can you see that? The waves becomes bigger. Right, balance, people, please, you must know. And this slide actually tells you everything. Firstly, if you change the direction and speed, the crista is being stimulated. And they are found in the ampulla, right? So if you remember acids, acid is for ampulla, which contains the crista. The I is just there to make it a word. For direction and speed, change in direction and speed. And this side, if you change the position of your head towards the gravity, right, then the receptor will be the macula. Where are we finding this? In the sacculus and the utriculus. Sump. So acids and sump will remind you. So when these things are stimulated, what do? They convert the stimulus into an impulse. And this impulse is then sent via the auditory nerve to the cerebellum. Just remember, in hearing, it's the cerebrum. In balance, it's the cerebellum, because cerebellum is balance. Right, so just this part is for both balance. This slide tells you everything about balance. Right. And that's the end of my section. The next section will be done by Ms. Kruger. who will join us in about 5 minutes. Thank you.
You are responsible for your own life. No matter how old you are responsible for the things that are currently happening in your life right now. Nobody is going to push you to become a better student. Nobody is going to push you to read more, to study more. You are responsible to put in the time necessary so that you can get the results that you want in your life. So today, make sure that you are putting in the work. Make sure that you are responsible in doing everything that you want to do. Because for you to achieve more, you need to put a little bit extra work to what you are doing and keep on believing. Keep on believing and keep on doing, and you will see the success that you want. I want you to ask yourself these 3 questions. Number 1, what do I want? Do I want to become an A student, or do I want to become an average student? Number 2, I want you to ask yourself, how do you plan on getting there? Are you going to put more extra hours, or are you going to just sleep? Number 3, who will be with you in this journey? Because it is important that we associate ourselves with students who have the same vision as us, who want to become a student, who want to put in the extra work, who want to become better at what they do. Unsuccessful people do not plan. They just let life happen to them. But successful people are planning on how they want their lives to be. And right now, you plan on how you want your results at the end of the year to be like.
Good day, grade 12 learners. Welcome back to today's Life Sciences lesson. My name is Radine Kruger, and I hope that so far you found this lesson is quite worthwhile, and you're getting ready for tomorrow's paper. All right, so what I'm going to carry on with is the other topics that are also in paper 1. And the first one I'm going to have a look at is reproduction in vertebrates. So if you have a look there, you can see it's only 8 marks. So it's not a very large topic, but there's mostly the concepts. That's what we need to know. So we need to understand the concept. So the first one is, or the first concepts that you need to know is the difference between internal and external fertilization. Then you also need to know the difference between ovipary, and vivipary. Right, then you also need to know the structure of the amniotic egg. You also need to know the difference between altricial and precocial development, and then what is parental care. Right. So let's have a look first of all at the different types of fertilization. The first one we're going to look at is external fertilization. Now, external is exactly what it says. The fertilization takes place outside of the body. So it's outside of the female body. There are advantages, but there are also disadvantages. So we're going to have a look at the differences. Right, so first of all, external fertilization means that there's no additional energy required, so very little energy is required for parental care or any kind of protective layer, for example, like the shell of the amniotic egg or any of those kind of things. So there's very little energy used by the parents. Secondly, there's no need for the male to have a special copulatory organ. So in other words, an organ that he uses to insert the sperm into the female's body. So there's no need for that because fertilization is external. And the third thing is that so that the female fish knows, or whatever it is, organism, the female knows that it's time for reproduction or fertilization or copulation or reproduction, they then have courtship behaviors. Okay, so they would have some kind of a display, either colors or, something like that, or a bit of a dance. Maybe they've got long tail fins or whatever, or they change color, and that alerts the female. Okay, the disadvantages of external fertilization is that the chance of the sperm cell and the ovum actually meeting for fertilization actually is very slim. Okay, because it's happening outside the female body, and remember, this is for organisms that live in water. Okay, external fertilization can only occur in water. So there's currents, there's predators, there's all sorts of things that can prevent the sperm cell from actually reaching the ova. Right. So that's the next point, is predators can eat the sperm cells or they eat the ova before fertilization takes place. There's also strong currents. We know that, in water bodies, moving water bodies, there are currents, and that can also wash or carry the sperm cells or the ova away from each other, and therefore reduce the chances of fertilization happening. All right, and then the other one that is very important, a very important disadvantage, and that's what I said. Remember, this type of fertilization is going to take place in water. So because it's external, it has to be in water, otherwise the sperm cells in the ova are going to dry out or dehydrate. All right, internal fertilization is the opposite. That is where the sperm cells are deposited inside of the female body, and so fertilization will take place internally. Okay, so there's a higher chance, what ensures that the sperm cells come into contact with the ovum or the sperm cell. Okay, so it increases the chances. And also, once when fertilization occurs internally, very often, or in some cases, the fetus or the egg is kept inside of the female's body. And so then she has protection. In ovipary, the fetus still, the developing fetus still has protection, because even though the egg is deposited outside the female body, it is encased in a shell, okay, which will then protect the developing embryo from harsh environment and things like that. The disadvantages are that there are fewer eggs or ova, and which would then be fertilized, that are produced. And remember, the other one is that the animal must have a copulatory organ. Okay, so it's unlike external fertilization. Here, the male must have a copulatory organ that he can insert the sperm cells into the female body. All right, now embryonic development, and those are the concepts of ovipary and ovovivipary. So the advantages, the eggs and the sperm cells, the ova and the sperm cells, are produced in large numbers to increase chances of survival to adulthood. Okay, and then also usually there is parental care. And then parental care also ensures survival to adulthood. Right, the advantages of these two, the young one is protected from the cold or from predators to ensure survival. They're also usually well developed, a lot more better developed. If you've seen organisms where external fertilization occurs, for example in frogs, the little tadpole is very underdeveloped, and that's why it goes through different stages of metamorphosis until it becomes a frog. Okay, so inside of the egg and inside, and when they stay inside the female's body, the mother's body, then they develop a little bit better. Okay, and that then increases their chances of survival. All right. Right, then vivipary, and that is the kind of development, embryonic development, we as humans also have. And that is the development occurs within the mother's body. So that ensures that the young are protected. Okay, so they're inside of the uterus. And Dr. Griselda went through human reproduction with you and spoke about the placenta and the amniotic fluid and how all of those have a protective role. Right, the young are born well developed and therefore less prone to be attacked. Parental care, we find in these kind of organisms, is increased parental care. So the parents then look after the young and ensure the survival, so that they then have a better chance of surviving to reach adulthood themselves. The disadvantages of vivipary is that we have fewer numbers of offspring produced. Okay, so depending on how large the animal is, that would depend on how many offspring they can produce. But if you look at cats and dogs who produce sort of largish numbers compared to humans, or other large mammals, you might find 7, 8, 9 offspring. But still, if you have a look at the fish and amphibians and those kind of organisms, we're large numbers, so that's a much smaller number. Okay, and then another disadvantage is there's a lot of energy that goes into looking after the young, protecting them, feeding them, keeping them warm. So there's a lot of parental care that is involved, and that uses a lot of energy from the parents. All right, so let's have a look at the amniotic egg. You need to know the basic structure, and we'll go through the different parts. So you see the embryo develops inside of the amnion. Okay, so there's an amnion as well in the egg, and that is why it's called the amniotic egg. And it's a membrane that is found surrounding the developing fetus or the embryo. And then there are other membranes. We find a chorion on the outside, and then obviously the shell. Okay, so it's a very hard shell. It's a calcareous shell, and so that is as part of the protection. All right, the amniotic fluid. So obviously the amnion has the same function as in viviparous animals, where we have the amnion secreting fluid, and that fluid then is called amniotic fluid, and that protects the fetus the same way, the same kind of functions, and also very importantly, it prevents it from drying out. All right, then the embryo inside of an amniotic egg, there we would find yolk, which is inside of the yolk sac, and that is the nutrients that is going to feed the developing embryo. And then also depending on how large or small the yolk is, that would then determine whether the animal, the bird that hatches out of the egg, whether it is precocial or altricial. So obviously the more nutrients, or the larger the yolk is, the better developed the little bird that hatches, or the hatchling will be. The less yolk, obviously, the less developed. All right. The allantois, that is another sac-like structure, and basically what that does is it removes, so it has an excretory kind of function where it removes metabolic waste. And then we have the chorion, as I said, is the outer membrane, and you can see it, that supplies oxygen, which allows actually the movement of nutrients or substances through it, so oxygen, food and water. All right, then let's have a look at the two concepts of precocial development and altricial. Okay, so precocial usually means, and if you have a look at this picture of the ostriches, you can see all those little baby ostriches there. They've got feathers, their eyes are open, they look like they're able to run around, they can probably pick on the ground, get themselves food. So this is what we call precocial development. So usually when the animals hatch, their eyes are open, their bodies are covered with soft feathers, they're able to move. Soon after they have hatched, they're able to feed themselves. They're also a little bit more independent of their parents. So, and you find this in larger animals that have precocial development, where the young need to, especially animals that might, be susceptible to predators. So those young then are able to run along with the parents. All right, then the other type of development is what we call, the young are highly developed, they're able to move and they're able to feed themselves. Then the other type of development we talk about is altricial. And this is where the young are not as well developed. So they are a little bit more helpless, and they require a lot more parental care. So the young ones are unable to move. They can't move on their own. Usually their eyes are closed. They don't have hair or they don't have feathers, and they're actually quite helpless. And they then need to be fed by the parents. They need to be kept warm by the parents, and they need to be protected by the parents. Okay, and for example, we are an example of that. Okay, it takes a while in our development as a baby before you are able to sit and move, crawl and walk, talk, feed yourself. Okay, it takes a lot of parental care. All right, so the immature individuals or organisms or offspring, they need a lot of parental care. All right, and then we've been talking about parental care, and that basically is the care that parents, when they look after the young, whether it be birds or any kind of mammal or humans, and that basically entails them feeding, looking after, protecting, keeping them warm, and that kind of thing. So, and then because it takes so much energy, parental care, usually the numbers where there is high parental care involved, the numbers of offspring produced at one time is a lot lower. Okay. All right. We're now going to move on to plant responses to the environment. And we're going to have a look at what we call plant hormones or plant growth substances. Okay, so we need to look at what we're going to focus on is we need to know the following 3 plant hormones, and that would be auxins, gibberellins, and abscisic acid. We must know the role of auxins in phototropism and geotropism, and then we also need to know about the protective mechanisms of plants. Okay, so plants are very clever. They can actually protect themselves from predators, from being eaten by predators. All right, so what are plant hormones? Okay, so remember we learned, or you've learned, and we'll have a look at it after this, is the human endocrine system. And hormones, we know, are chemical substances that are produced in one part of the body and then they function elsewhere. However, with plants, it's different. So they are not regarded as true hormones because they work in the part of the plant where they are produced. Okay, so therefore we more refer to them as plant growth substances, and that is because they stimulate or they inhibit growth. Remember, a plant, if it feels like it's in too shady a spot or there's not enough sunlight, it can't lift its roots up and move to a sunny spot. What it has to do is it needs to grow towards the part or the area where there is more light. Okay, so that is why we talk about it as a growth substance. Okay, and what these do, these allow plants, so these growth substances allow plants to respond to certain stimuli in the environment. And the type of growth movements that we look at, we're going to look at tropic movement, and that is a response specifically to the direction of the stimulus. Okay, and so what these substances then allow plants to do is they allow them to bloom, to flower at the right time. We don't want flowers developing in winter, okay, because then there probably won't be pollinators around. It might be too cold, the conditions are dry, so they must bloom when the conditions are favorable. So that regulates that. Then they also allow plants, as I said, to grow towards the light stimulus, because remember, light is very important for photosynthesis. They also allow seeds to germinate when the time is right, so at the beginning of spring, and again not during winter, because remember, fruit and seeds are formed at the end of summer, but they don't germinate straight away. They remain dormant through winter. And so there's also a hormone that induces dormancy and prevents the seeds from developing in winter or germinating in winter, and rather keeps them dormant until the conditions are favorable. All right, so let's have a look. All right, so we're going to have a look at the 2 abscisic acid and gibberellins. Now, the first one, abscisic acid, acts as an inhibitor of growth, so it inhibits or slows down or stops the growth. So it causes the plant to become dormant in winter, because remember, in winter the sun, the temperature is a lot lower, there's less moisture in the air, in the soil, so there's not a high amount of sunlight either for photosynthesis. So for the plant to maintain a normal or a metabolic rate where it's growing during summer, there's not enough nutrients. So they then become dormant in winter. Also, abscisic acid is also important in that when there's a drought, so whether it doesn't matter what time of the year, when there's a drought, it causes, or dry period, then it causes the stomata to actually close. Because remember, when stomata are open, they allow the exchange and the release of water, so transpiration increases, or the rate of transpiration increases. So what it does is it causes those stomata to close, and in that way it prevents water loss. Right, then the next hormone that you need to know is gibberellins, and gibberellins is the hormones that then encourage stem elongation. So when your plant grows taller, and it's important that your plant grows taller because it needs to grow to the sunlight, it also then stimulates the germination of seeds. So around this time of year, when it's spring and conditions are starting to warm up, we would find a high concentration of gibberellins in the plant. It also promotes flowering. All right. Yeah, and we can use gibberellins. Obviously, we can use it when we want to make things larger, or we want organisms or plants to flower early, or promote growth, stimulation of seeds and things like that. All right, so there you can see that diagram, or that picture shows you there where the abscisic acid has caused the open stoma to actually close, so that we have less water loss. Right. Okay, now we're going to have a little look at in a little bit more detail at auxins. And basically, auxins have a lot of functions, have many functions. So they stimulate, here we can see what they do is they stimulate cell division, cell elongation. So cells divide, and then they're also going to grow. So it causes growth in the stem length, for example. Also, it causes the development of fruit, abscission of leaves and fruit, so that's when they fall off. The development of adventitious root systems. So if we take a plant cutting and we put it in some auxins powder, rooting, we call it a rooting powder, it will cause that leaf structure to actually develop roots, and then we can plant it and grow a new plant. That would be vegetative reproduction. It's also responsible for tropic movement in stems and roots, and it also ensures apical dominance. So in other words, it's produced in the apical bud, which is the bud right at the tip of the stem, and it encourages elongation rather than the plant bushing out. And sometimes that's very important for conditions that the plant might find itself, is that shape of apical dominance. Also remember, if there's lots of competition and lots of other plants around, then the plant would like to grow tall to get to the sunlight so that photosynthesis can occur. All right, so if we have a look at the diagram here, we can see there's a plant shoot. That's what we call a shoot. It suppresses the growth of lateral buds. Lateral is to the side, so it suppresses that, and it causes the stem to grow upwards. So we don't have sideways growth, a lot of that. Okay, it's more growing longer. It causes cell division, so that's going to be growth. It also causes elongation, and if you have a look here, you can see those cells have grown much longer than the ones on this side. Okay, and that then causes tropic movement, where the stem is going to grow towards, because we can see we have a unilateral light. Okay. All right, so let's have a look at phototropism and geotropism. As I said earlier, tropism is a growth response which a plant makes with respect to a directional stimulus. So it's going to grow towards a stimulus or away from it. Okay, so they can either be positive, okay, where we see here positive, that is when it's growing towards the stimulus, and negative is when it grows away from the stimulus. So if we say that stems are positively phototropic because they're going to grow towards light, roots don't want to grow towards light because there's no water in the air. They need to grow in the soil where the water and the nutrients are, so they would be negatively phototropic. They grow away from the light. Okay. Whereas if we have a look at geotropism, roots are going to grow towards gravity because that is where the water is. It's in the soil. And stems, they need the light. They can't grow underground because then they're not going to get any light. So it's very important that they are negatively geotropic. So they're going to grow away from gravity. All right. Okay, so what we have here is an investigation and examiners like to make or to have an investigation question. So here we can see some apparatus. This is a box, and what we've done is we are demonstrating phototropism in this first investigation. And what we see here is we can see that there's a hole in the box and there's light coming through on one side. However, in the experiment, this pot plant has been placed on a stationary clinostat. A clinostat is a piece of apparatus that when you wind it up or you switch it on, if it's electronic, it rotates very slowly, so it turns. So in this case, we don't switch it on or we don't wind it so that the plant stays stationary, which means it's only going to receive light from one side. Whereas in this set of apparatus, and we call it the control because we are eliminating the variable that we are investigating, so that clinostat is then switched on or wound up. So it's going to move. And when it moves, when it turns like this, the plant is going to turn with it because it's on the station, it's on the clinostat, and that means the plant is going to be receiving light from all directions. Okay. Then another investigation or another set of apparatus or a way in which we can use the apparatus to set up an investigation is to investigate geotropism. Again, we have a clinostat, and what we have done here is they've placed bean seedlings on the clinostat. In the control, again, we have eliminated the one-sided stimulus, and we are now ensuring that the bean seedlings are exposed to stimulus from all directions. Whereas in this one, it's a stationary clinostat, so it's not moving. So remember, roots are going to grow towards gravity and the stems are going to grow away from gravity. And that's exactly what we can see in this diagram. Whereas these ones will grow, carry on growing in the direction that they were placed on the clinostat because they are getting the stimulus from all sides. All right, so those are the things you need to know. You need to know what a clinostat is. And remember, when we set up a control increases the validity of the investigation because what we do is we set up a second set of apparatus and we eliminate the variable that we are testing, so the independent variable. We remove that to see if the results are caused by that specific variable or if something else might have affected our results. Okay. Right, now if we have a look at the role of auxins and we look at the differences. Okay, so they're slightly different in the root. They act or they function differently in the root and the stem. So auxins in the stem are produced at the stem, so usually in the apical meristem. And whereas in geotropism, auxins are produced at the tip of the root. Auxins move downwards in the stem, whereas they'll move upwards in the root. And then they are evenly distributed if the stimulus comes from all around them. And that then makes for even growth. Okay, so we won't have a bending in any way. So the stem then will grow upwards and the root will go downwards. However, when we have a unilateral stimulus, in other words, a stimulus that comes from one side, one's lateral side. So in phototropism, when the stem is exposed to unilateral light or light from one side, auxin concentration, so remember it's produced at the tip of the stem. So what happens is if that's where the light is on this side, then the auxins are going to travel down the side away from the stimulus or the darker side, okay, and that is because auxins are destroyed by light, so they move to the other side, and what they do there is they encourage or stimulate growth. So these cells are going to grow much quicker and larger, and that causes the stem to bend towards the light. Now in roots it's slightly different, okay. So if a root is placed horizontally on its side, what happens is, again, at the tip of the root, the auxin is going to be produced, but it's going to spread down the underside, the bottom side of the root, because gravity is going to attract it down to that side. And what it does in the root, it inhibits growth. So in other words, these cells on the underside are going to grow slower than these ones at the top. And then what it's going to do is it's going to cause the root to bend downwards towards the water. All right. Okay, let's quickly look at the role of chemicals and thorns. In this picture here, you can see these are thorns that we find on roses. In some plants, we have very long, sharp, and hard thorns, lots of thorns, and they then act as protection so that herbivores don't eat the leaves. And then we also find chemical defenses. So a lot of plants produce substances in their leaves, such as tannins, like we find in tea leaves, cyanide, those kind of things, and they actually make the leaves taste bad, or they are actually poisonous, and they can cause, an organism to die. So that is how plants then protect themselves from being overgrazed by predators or animals, herbivores. All right. So let's quickly look at the human endocrine system. As you can see, it's quite a large section. There's 34 marks. So basically what we need to know in the endocrine system is the structure of it, the different glands, the hormones they secrete, the target organs for those. We also need to know what a negative feedback system is. And in homeostasis, we must know basically what homeostasis is. And very importantly, we must be able to explain how our body regulates glucose, how it regulates carbon dioxide, how it regulates water and salts, and then even our body temperature is regulated in homeostasis. All right, so quickly, if we have a look here, we can see the many glands. And if you have a look here, first of all, that's the hypothalamus, which you know is part of the brain, but it does produce ADH. However, the hypothalamus doesn't secrete ADH. It doesn't send it into the bloodstream to go and work at the kidneys. What it does is it stores that ADH in the pituitary gland, and it's the pituitary gland that actually secretes it when it is required. Okay, so then the pituitary gland, there's a few hormones that are secreted by the pituitary gland. You learned about growth hormone, you learned about thyroid stimulating hormone, and many of these hormones, their names tell you exactly what they do. It also secretes FSH, or follicle stimulating hormone, luteinizing hormone, prolactin, so a couple of those hormones. All right, then the next is the thyroid gland. So as we're moving down the body, the thyroid gland, we know, produces thyroxine, and thyroxine regulates our metabolism. Then if we have a look at the pancreas is very important. It secretes two hormones, insulin and glucagon, and those are then used to regulate our blood glucose to make sure that it's balanced. You don't have too much or too little. The adrenal glands, also very important, they secrete adrenaline, which works very much in the same way as your autonomic nervous system, as your sympathetic nervous system, that gets you ready for an extraordinary situation. So tomorrow morning, perhaps before you write your exam, you're a little bit nervous, and that is initially, first of all, your sympathetic nervous system, and then adrenaline kicks in and it gives you those butterflies while you're waiting, while the invigilators are handing out your papers. All right, and then the adrenal glands also secrete aldosterone, and then the ovaries, they then produce the two hormones, estrogen and progesterone, which you learnt about. And then in males, that's in the females, and in males, the testes is going to produce testosterone. All right. So the negative feedback mechanism, if you have a look in Mind the Gap, there's a sequence of events or 7 steps, where the first one is your body detects an imbalance. Okay, so something is not balanced. That then stimulates the control center, whether it be your brain or whether it be the pituitary gland or whatever. So it's stimulated, and then the control center is going to respond. So once it gets the information that something's in imbalance, analyzes that information, and then it works out a response. Okay, and what it does is it then sends the message, okay, to the target organ that is going to bring about the change. And that change, hopefully then, when the target organ responds to bring about that change, it's then going to reverse the imbalance. So where we had some too high levels or too low levels, hopefully the levels now come back into balance. All right, that's step 7. Right, now what's very important to know that in a negative feedback system, there are 3 components. You must have a receptor. Okay, and remember that's our receptor or our sense organs. And we don't only have those ones that we know, our eyes, our ears, our nose, our tongue. There are others that are sensitive to other stimuli. So we have a receptor, which is then takes the information to the control center, and the control center is going to analyze and integrate that information and say, okay, this is how the body must respond. And the response is going to be brought about by the third part, which is the effector. So we've got the receptors, number 1, the control centers, number 2, and the effector is number 3. All right, so let's have a look here quickly at the levels of thyroxine in the human body. Okay, so here we see the thyroxine levels have increased, or either if they increase or they decrease. So here we've got high levels of thyroxine in the body. So basically it's above normal levels. So your pituitary gland is stimulated because it gets that information. And what it does is it reduces the amount of TSH, which is thyroid stimulating hormone, that it is secreting. So the thyroid gland is stimulated less, which means it's not putting out as much thyroxine. And so that levels of thyroxine then are going to decrease, which then brings it back into balance. It returns to normal levels. All right, and exactly the same is when the thyroxine levels are too low. Again, your pituitary gland will be stimulated. It's going to increase its amount of TSH, which then would stimulate the thyroid gland more frequently, which means more thyroxine, and hopefully those low levels are going to be raised to normal. Right, now when we have abnormal functioning of the thyroid gland, we call it goiter. It's known as goiter. And you can see here in the picture that thyroid gland, which is in your neck and which you can't actually see, can become quite enlarged. Okay, you can see this is a very enlarged one, and it's often linked to overactive thyroid in the sense that it's doing a lot of activity. However, it's not necessarily producing thyroxine. Okay, so there are symptoms, and that's an increased metabolic rate, increased cardiovascular activity, increased anxiety because you're going to be very, on edge, and it can also, it doesn't always lead to a swollen thyroid, but it can. Okay, so what you need to know is we learn it as a negative feedback. So what happens is the pituitary gland gets the message. So we start over here that the thyroxine levels are low. So that's the feedback that the pituitary gland gets. So what's the pituitary gland's response is to increase the amount of TSH in the blood. Now normally the TSH would cause the thyroid gland to produce more thyroxine, and then the thyroxine levels would be brought back to normal levels. But here, in this case, the thyroid gland is not producing thyroxine for whatever reason. So it continues, or the feedback that keeps going back to the pituitary is that there are low levels of thyroxine. So it's continually, that feedback mechanism is continually more TSH, which then tries to stimulate. So the thyroid gland is really working hard. But it's not managing to secrete thyroxine, and that leads to goiter. Right, the negative feedback mechanism of glucose. So here we should have normal levels of blood glucose. And so when the glucose levels increases, and it's usually after we've had a meal that is high in carbohydrates, so our glucose levels in the blood increase, and that message eventually gets back to the pancreas. Pancreas is then stimulated to secrete insulin. Insulin then goes via the bloodstream to the liver and the muscle cells where it functions, and what it does is it encourages the liver cells and the muscle cells to absorb glucose from the blood. Once that glucose is into those cells, it's then converted. So it's built up into a carbohydrate, which is glycogen in animals. All right, so then it then causes the glucose levels in the blood to decrease. Now, if the blood glucose levels go too low, then we're not going to have enough energy. So once those glucose levels then decrease below normal levels, then the pancreas is stimulated again, and it secretes the hormone glucagon. Now, it's very important for you to understand or remember to spell these correctly. Glucagon is the hormone, glycogen is starch, is stored starch. Okay, so don't mix the two up. All right, so as soon as our levels, as glucagon is secreted, it goes via the bloodstream again to the liver cells, and it stimulates those liver cells to break down that starch that it had built up, now the glycogen that it had built up. And that then gets broken down into glucose molecules again, which can move, or they can diffuse through the membranes back into the bloodstream, and that's going to increase the glucose levels. All right, same, we also have a negative feedback mechanism with our water and salt balance. Remember, you did study this last year. So again, we're going to have to control our water balance in our body. The hormone that is released is ADH, and it's the pancreas that releases it. And that then goes to the kidneys, and it makes the kidney, the tubules of the kidneys, more, that they allow more absorption, so that water is then, they make them more permeable, and that allows water to be drawn back into the blood. That is then going to increase the levels of water. If we have a lot of water in our body, okay, we need to get rid of water, then antidiuretic, which is then going to, we're not going to have as much, so there would be less ADH that is secreted, so that our bodies can get rid of, so we produce more urine and get rid of that excess water. All right, and then salt regulation is also very important, because remember, when there's a lot of salt in the blood, it's going to cause water to accumulate because it wants to keep an osmotic balance. So this, when the salt levels are high, it's going to cause aldosterone, or it's not, there won't be aldosterone release, because what happens is you want that the salts then to be deposited in the filtrate of the kidneys so that we can get rid of it. And when our salt levels are too low, it's aldosterone will be released, and then that again causes salts to be absorbed back into, or reabsorbed back into the blood, to increase the salt levels in the body. Okay, then going to have a look at thermoregulation quickly. So you know that your skin plays a very big role in regulating your body temperature. When it's hot, we're going to have the blood vessels that run underneath the surface of the skin. There you can see they're going to dilate, and remember, when we dilate, it becomes larger, and that's going to allow more blood flow. And when there's lots of blood flowing through there, it's going to increase the activity of the sweat glands, and the sweat glands are going to make more sweat, which is deposited on the skin. And it also, with that hot blood running, lots of hot blood running through just under the surface of your skin, it's going to give off heat. So we're going to lose heat. It's lost through heat, evaporation and radiation. Okay, so that is how we're going to lose heat. when we get too hot. And then when it's cold, what happens is when it's cold, then we don't want to lose as much heat because our body temperature will drop. So what happens there, you can see those blood vessels again that run under the skin, they're going to constrict. And when they constrict, it means less blood is flowing, which means the sweat glands are going to be less active. So we don't sweat as much when it's cold. Also, our skin goes colder, so that we then give off less heat. Okay, and that process we call thermoregulation. All right, things to take into the exam room. Remember, you're going to possibly be asked in one of the papers and maybe in prelims in both of them. You will be asked to possibly draw a graph, so you need a ruler for sure. You may be asked to draw a pie graph, which means that you need a compass and a protractor. You definitely need a pencil and an eraser so that you can draw the graphs, and if you make mistakes, you can erase. You need a calculator because more than likely you will be asked to do a calculation. And then you need pens, preferably a blue pen, and then make sure you have a backup so that if your one pen stops writing, you will then be able to carry on. All right, now we want to have a look at some of the questions that we have based on the topics that we've covered. And if you have a look at the first one, the question says, describe the process of spermatogenesis in part A. So if we have a look at this diagram, we can see that's part A, which is the testis. And remember, Dr. Griselda said 4 lines in the exam guideline, you must know them. So if we have a look here, we can see that is what is on the memo. So please learn those lines in the exam guideline as they are, because it says under the influence of testosterone, diploid cells in the seminiferous tubules of the testis undergo meiosis to form haploid sperm cells. All right. Here again, the next question says, test results show that a man has a low sperm count. Explain why a doctor would advise the man to wear underwear that is not tight. So remember, that is why the testes are found in the scrotum, because the scrotum can regulate the temperature, and normal body temperature is too warm for the normal production of sperm cells, and that is why, so if the male is wearing tight underclothes, it's going to hold the testes too close to the body, going to increase the temperature, and that then might not or would affect the production of sperm. Okay, so the testes will be further away from the body if the underclothes is not too tight. Temperature of the testes therefore will be lower than body temperature for successful sperm production. Or you could answer it the other way and say tight underwear pulls the testes closer to the body, increases the temperature of the testes, which then will affect the maturation of the sperm. Okay, and so they would be negatively affected. All right, then if you have a look here, this is a diagram that shows the ovarian cycle as well as what is happening in the uterus. We can see over here the lining of the uterus as it gets thinner and then as it's being built up. And here we see what is happening inside the ovary. So the first question is, they say name hormone V. So if we have a look, there's hormone V. And we know what increases the thickness of the endometrium, and that would be estrogen. So even though it's spelled with an O, we pronounce it as estrogen. And then describe what effect V has on the lining of the endometrium. So basically what estrogen does is it encourages the development. So the endometrial lining becomes thicker and more vascular. Okay, it increases the thickness. And explain the importance of the endometrium lining. Well, that is the part where implantation is going to occur. So that's where it implants, and then it's going to form the placenta. And remember, the placenta is a very important connection between the fetus and the mother. Okay, Then they ask us to name hormone X. So there we can see, and here we see that they name that organ as the pituitary gland. And we can see hormone X is causing ovulation. It's also caused the follicle to change from the Graafian follicle to the corpus luteum. So that must be luteinizing hormone. All right. And then the functions of hormone X, as we've mentioned now, it's produced to burst the Graafian follicle or to release the ovum or stimulate ovulation, and it stimulates the development of the corpus luteum. All right, then we have a look for hormone Y, and we can see hormone Y over there, and they say which hormone, and it's not hormone, sorry, structure Y, so that is this structure there, and the hormone that is produced by the corpus luteum would be progesterone. Okay, and then the two functions of this hormone, again, it functions here to thicken, to continue the thickening of the endometrium. So it prepares it for implantation. It maintains the endometrium so it doesn't tear away and isn't expelled as menstruation. So it keeps that uterine wall, the lining, in place, especially if fertilization and implantation occurs right through the pregnancy. And then its other function is, if there's high levels of progesterone in the blood, that message goes to the pituitary gland in a negative feedback mechanism, and it inhibits the secretion of FSH, because if the female is pregnant, there's no need for new follicles to develop. All right, then hormone Z. Here we can see hormone Z coming from the pituitary to one of these immature little cells or structures, and those would be the immature follicles. So hormone Z would be follicle stimulating hormone, or FSH. One of the functions is to stimulate the growth or development of follicles. And what will happen to the levels of hormone Z in the bloodstream if the level of hormone secreted, so if it's high, I would assume. Okay, so if there's a high amount of progesterone, as we said earlier, it then will cause less. So we would have less FSH in the blood. All right, it will decrease. Okay, quickly look at the human nervous system. The retina is the light sensitive layer of the eye. So we can see that's the inner lining here of the eye. And they're asking us specifically here where the optic nerve leaves the eye, why no image can form in C, and C we know is the blind spot. So at C, there's no photoreceptors, so we don't have any rods or cones, and therefore no vision occurs. All right, then with reference to the structures labeled A and B, so here we can see B is the lens, A is the suspensory ligaments. Explain the accommodation that will take place when a person is viewing an object close to him, so they're looking close up. So what happens then is the suspensory ligaments are going to slacken, okay, which means that our lens is going to become more convex, right? We can't say that it is rounder, so it decreases the tension on the lens, and then that makes it, it becomes more convex, and then increases the refractive power of the lens so that we can see close up. Right, the ear. If we have a look at the different parts here, they ask us quickly to give one function. So A would be the external auditory canal, and the function of that is to transmit sound waves. Let's get the answers. It transmits sound waves to the tympanic membrane here. It also secretes earwax, which is protection. Okay, and then structure E, which is our eustachian tube, and that basically regulates the pressure on either side. It will equalize the pressure on either side of, so that the pressure stays equal on either side of the tympanic membrane. So if we have an increase on one side, it's going to cause the membrane to rupture, whichever side has the higher pressure. And then structure F, which is the round window, and basically what that does is it releases the pressure waves or the the waves, the stimulation of the waves, so that we don't have ringing in our ears. It allows the sound to dissipate. Okay. Explain why the fusion of part B may lead to hearing loss. So part B is our 3 little bones that we find in the middle ear. If they fuse, then they're not going to be able to vibrate against each other. And remember also another important function in the middle ear is that because the tympanic membrane and the oval window it's much larger than this one, is much smaller, it causes amplification. So if those bones fuse, then it's going to prevent the transmission of those vibrations from the tympanic membrane to the oval window, and also no amplification will occur. Okay, so they won't be able to vibrate, no vibrations will be passed on to the inner ear, and no amplification, and that obviously would negatively affect hearing. All right, explain why people with middle ear infections are usually advised not to travel by aeroplane. So remember, when there's middle ear infection, it usually means the eustachian tube is blocked so that it is unable to equalize pressure. And when you go high up, the pressure on the outside of your ear, so from the outside, will be a lot lower than what is inside. And because it can't equalize, it's going to cause the tympanic membrane. It can cause it to rupture. Okay, so mucus in the middle ear from the middle ear infection will lead to blockage of the eustachian tube, which will not be able to equalize the pressure on both sides of the tympanic membrane, resulting pressure on the tympanic membrane that may cause it to burst, okay, which will then lead to hearing loss. All right, then quickly on plant responses. If we have a look at this diagram, we can see the sun is on one side, so the shoot has grown towards the stimulus. So the growth response here is called phototropism because it's in response to the light. The hormone that is responsible for this growth response in the shoot is auxins. And then on which side, A or B of the shoot, will the hormone accumulate most? Remember, we said the auxins are going to be produced here at the tip, and they're going to move down the side that's away from the light, because light destroys them. So we would find them more on side B. Okay, they would be higher on side B. All right, then some terminology, and terminology is very important. You must learn it. So the first one, they give you the description and they say, write only the term. So if we have a look at the first one, it says substance that stimulates cell elongation on the shaded side. So that would be auxins. Plant substances that act as a growth inhibitor. Abscisic acid, remember we said that makes the plant become dormant in winter. Movement of a part of a plant in response to a stimulus, that is tropism. And then the tissue in which the auxins implant is mainly produced, and that would be meristematic, apical meristem or meristematic tissue. Right, and then quickly at homeostasis, if we have a look at this diagram, we can see here this represents the epidermis of the skin and the dermis. These would possibly be blood vessels, and here you can see they say blood vessels under normal environmental conditions. Here we can see the blood vessels are much smaller, and then here we see the blood vessels are much larger, and you can see here we see that there's more of something being released. Here there seems to be less. Okay, so they say describe the environmental condition at X. So if the blood vessels are constricted and we're releasing less heat, then that must be cold conditions. Okay, what is represented by the arrows? That would be heat being released, heat loss or radiation, and the process that is represented in the diagrams that maintains a constant body temperature, and that would be thermoregulation. Right, and then this question, it's a little bit of a longer question. You can see it's worth 4 marks, so we must read carefully. Here they say describe the negative feedback reaction that occurs in the body after the brain has detected a person is hydrated and the blood has a high water level. Okay, so our water levels in our body are high, they're above normal levels. So the body is going to respond, and that means that less ADH is secreted, because remember, ADH is prevents the loss of water, so it makes your body hold back water. So less is secreted by the pituitary. The kidney tubules become less permeable, so the water is going to stay in the filtrate inside of the nephron, and so less water is reabsorbed into the bloodstream, which then causes the amount of water in the blood to drop. So the levels decrease, blood volume is going to decrease. More water is going to be excreted as urine, and also the urine becomes less concentrated, and so hopefully in that way our water levels will be brought back to normal. Right, and that is the end of our lesson for today. I wish you guys a lot of luck and a lot of hard work that you're still going to do tonight, and remember, hard work always pays off. So please prepare yourselves well today and tomorrow, for paper 1 tomorrow and for paper 2 on Monday, and I hope this was helpful. Thank you.